Hi, I am Sudip Mondal, a postdoctoral fellow with Dr. Sandhya P. Koushika at National Center for Biological Sciences, India. In this video, I am going to show you how to fabricate a bilayer microfluidic device using soft lithography techniques in a regular lab environment. I am going to use these devices for high-resolution cellular and subcellular in vivo imaging of genetic model organisms. The current protocol will allow researchers to perform this time-lapse imaging in anesthetic-free intact organisms. So let's get started. We have developed a simple microfluidic device to perform anesthetic-free in vivo imaging of genetic model organisms such as C. elegans, intact Drosophila larvae, and Zebrafish larvae. In this study, we demonstrate a membrane-based immobilization device fabricated using photolithography of SU8 and soft lithography molding of PDMS. In order to immobilize single organism, we locate